To finish up our series on antibiotics, we'll now take a look at drug resistance. Drug resistance is not something new. It's been around for a long time and we've known about it. We know organisms evolve to their environment. And in fact, when Alexander Fleming first discovered penicillin, he had a fear that in the future penicillin would be so overused that eventually microbes would become resistant to it and we wouldn't be able to really use it anymore. And sadly, his premonition about that has come true. What happens with antibiotic resistance is that it applies what we refer to as selective pressure on the organisms. Microorganisms that have the ability to overcome these antibiotics or withstand these antibiotics then can pass those genes on to one another. And as we've seen from our study of DNA and RNA and chromosomal material, the bacterial and eukaryotic genomes, we know that these organisms can swap and exchange DNA. So if a particular microorganism has a resistance plasmid that contains information on how to resist an antimicrobial drug, they can spread it. They can pass it along so that we get whole populations of microorganisms that are now resistant to a particular drug. Now, what are some of these factors that can exacerbate the problem? Number one, overuse of antibiotics. We know that there are a lot of viral illnesses out there that antibiotics, antibacterials, are completely useless against. Yet when people go to the doctor and they're sick, they want their doctor to give them some antibiotic that's going to make them feel better. And the fact of the matter is, not all illnesses can be treated with antibacterial or antimicrobial agents. Misuse of antimicrobials. And this goes back to, prescribing an antimicrobial for a viral illness. Inappropriate use, using them off-label, using them for things that they're not designed for. Subtherapeutic dosing. What this means is prescribing a dose that is really too low to wipe out the entire infection. And then patient noncompliance. Not taking the medication as specified, not completing the entire course of the antimicrobials, stopping treatment when they feel better. So there are a lot of factors that have really sped up this process of drug resistance. So when pathogens are exposed to some type of antimicrobial agent, this can turn genes on that they may not have ever used before. If they've never been in the environment with a particular antimicrobial agent, then that gene may have never been needed. It may not have ever been turned on. So it's just kind of sitting there hidden. And then when they're exposed to that particular drug, then that gene turns on and confers resistance, and then that microbe can pass that on to subsequent generations or to other microbes. So if we look at this image here, we see some of these mechanisms for drug resistance. So what are some things that can happen? Number one, blocked penetration. So the microbe can actually prevent the agent from even getting inside the cell. If it can't get inside the cell, then it can't work. So some organisms have mechanisms that block the drug's ability to get into the cell. What's another one? Target modification. They can use their genes to actually modify the target of the antimicrobial agent so that it can't attach and work as it needs to. Inactivation of enzymes is another. A perfect example of this, would be like the beta-lactamases, and then an efflux pump where the drug may get in, but then there's a pump that pumps it directly out so that it can't do what it was intended to do. So in this next section, there's a little bit more information about each one of these things. Like the target modification, if the pathogen changes the target structurally, then the drug can't bind to its target and it can't do its job. And this could be through spontaneous mutations in their genome. And you can see there some of the examples. Ribosomal subunits can change. The lipopolysaccharide on its cell membrane can change. RNA polymerase can change structure. So these organisms can really outsmart a lot of different types of antimicrobial agents. So looking at a few specific multi-drug resistant microbes and cross resistance. So these are our greatest concern. When we have pathogens that are resistant to one or more antibiotics or antimicrobial agents, it really poses a concern, not just to the patient, but to the medical community as a whole. Because we know that if these types of resistant bacteria get out into the general population, then something that was easily treated 10, 15, 20 years ago could now prove fatal. These multi-drug resistant microbes we tend to call superbugs. They carry one or more of those resistant mechanisms. And so if we look at some of these, CDC reports that superbugs are responsible for more than 2 million infections in the United States annually, resulting in at least 23,000 fatalities. It's really sad to think that we developed all of these antimicrobial agents, and for so long, bacterial and pathogenic illnesses were the #1 killer of man because we had nothing to treat them. And then we developed all of these antimicrobial agents, and now we're finding ourselves going back. We're moving backwards. We've hurt ourselves by not using antimicrobial agents in the right way. And this is, leading us down a path that we don't really want to go down. So some of the superbugs you see there have been assigned this acronym ESCAPE. Enterococci faecium, Staph aureus, Klebsiella pneumoniae, Acinetobacter, Pseudomonas aeruginosa, and Enterobacter species. So these pathogens can prove to be difficult to treat. So the first one we look at here is Mrsa. This is probably one of the first that we were really know about, like that we learned about, that we understood was out there and has been, extensively studied. Methicillin is a semi-synthetic penicillin. So we know penicillin is a true antibiotic because it comes from Penicillium mold. Methicillin is a semi-synthetic, and it was designed to be able to resist inactivation by those beta-lactamase enzymes that bacteria can produce. However, after we recognized that, normal penicillin was not killing some strains of Staphylococcus aureus anymore, we started using methicillin, and it didn't take long for there to pop up resistant strains to methicillin. And Staph aureus can now pretty much have resistance to all of our available beta-lactam medications. At first, we kind of just saw this in healthcare settings, what they refer to as HA Mrsa, but now we really are seeing it in the community, which is the community-associated or CA Mrsa. You see there about a third of the population carries S. aureus around in their nose without it causing any problems, and it's believed about 6% of those strains are methicillin resistant. So as long as they stay in the nasal cavity, they're not causing any problems. It's where they get, into areas where they don't belong, especially under the surface of the skin, that they really start causing problems. So then we started using vancomycin to treat the Staphylococcus aureus infections that were resistant to penicillin and methicillin. And what popped up? Well, now we have Mrsa, vancomycin resistant Staph aureus, and Visa, vancomycin intermediate Staph aureus. We also have vancomycin resistant Enterococci. And the problem with this is vancomycin is essentially almost our last line of defense. We do have the linezolid group of antimicrobials, and we haven't seen resistance to that yet. That's been documented, but we're really getting to the end of the line in terms of antimicrobials that can treat these infections, these superbugs. We also have carbapenem-resistant Enterobacteriaceae, and carbapenem, again, is one of our last lines of treatment. And then we have MDR and XDR TB, which we'll talk a lot more about when we talk about tuberculosis, but this is multi-drug resistant and extensively drug resistant Mycobacterium tuberculosis. We don't see as much of this in the United States. We do have some of these strains here, but mostly we see this in areas where there is poor access to health care. This can lead to huge problems in individuals who have co-infection with HIV and TB, because if they acquire one of these resistant strains of TB and their immune system is already weakened because they have HIV or AIDS, then we're really running out of treatment options for these individuals. So we'll talk about a few of these in a little bit more detail as we go through our pathogens here over the next month or so. The key thing to remember about all of this is don't ask for antibiotics from your doctor if it's a viral infection. If you do have a bacterial infection or other type of microbial infection that does require the use of some type of antimicrobial agent, take it exactly as directed and take it until it is all gone. Make sure that you follow the directions and you do exactly as specified. You don't want to develop a superbug that isn't going to be treated with normal antibiotics.